is administered directly into the circulation systems. In this video, there are four main topics that are being discussed. Firstly, the indication and the formulations that are available in order to produce this product, whereby 1% propofol is formulated into the 10% soybean oil emulsions with the additions of other excipients.
The sterile manufactured product of the Propofol Emulsion injection also is discussed. The product is manufactured in aseptic process under the laminar flow with Class A and Class C environment as the background. This is because the product is not suitable to be used under the heat sterilization and radiation sterilization for some reasons. However, the container that is being used for packaging can be used under the dry heat sterilization.
Next, quality control tests that are being conducted according to the specifications of the product are being explained further to know the extent of the product to be accepted or rejected before being marketed.
Lastly, this video briefly explains about the administration step of the product and also the precaution that should be taken before administering the product so that contamination can be avoided.
Now, let's take a look at the formulation of Propofol Emulsion injection. What is the ingredients included in the formulation of Propofol Emulsion injection? Based on Handbook of Pharmaceutical Manufacturing Formulations of Sterile Product, the formulations include propofol, soybean oil refined, egg lecithin, glycerin, sodium hydroxide, water for injection, and nitrogen gas NF. In this formulation, propofol is the active pharmaceutical ingredient and all of the other ingredients are non-active pharmaceutical ingredients. The function of soybean oil is to hold the bulk of the propofol. This is because propofol emulsion injection is formulated in a medium that is possible to be stabilized and dispersed. Next, egg lecithin acts as an emulsifier as it functions in stabilizing the small droplets of propofol soybean oil in aqueous dispersion. In addition, glycerin helps to maintain the pH of the formulation to be isotonic with blood. What about the indications? According to the FDA, there are 5 main indications for this drug. For adults, this drug is used in 3 conditions. First, initiation and maintenance of monitored anesthesia care sedation. Second, combined sedation and regional anesthesia. And third, intensive care unit sedation of intubated mechanically ventilated patients. Next, for patients greater than or equal to 3 years of age, this drug is indicated for the induction of general anesthesia, whereas for patients greater than or equal to 2 months of age, this drug is indicated in the maintenance of general anesthesia.
Manufacturing sterile emulsions can be quite tedious and costly. Conventional sterilization, such as heat sterilization, is not suitable because it can affect the stability of the thermolabile materials. Meanwhile, the radiation also is not suitable because it can result in the production of the free radicals, which can promote degradation of the excipients. So, what is the suitable method that can be used in order to produce these drugs?
Since heat sterilization and radiation is not suitable, propofol emulsion injection is sterilized through aseptic technique. In aseptic technique, the drug product, container, and the closure are sterilized separately and brought together under aseptic conditions. The aseptic process is conducted under laminar airflow in a Class A environment with a Class C environment as a background.
So, how the drug product is prepared?
The drug product or the emulsion is prepared by mixing the oil phase and the aqueous phase. The aqueous phase is prepared by mixing glycerin and the lecithin in the water for injections until a uniform dispersion is formed. Meanwhile, as for the oil phase, it is prepared by adding propofol to the soybean oil and stir until dissolved. The oil phase is mixed with aqueous phase and the primary emulsion is formed. The preparation of primary emulsion is conducted at temperature of 40 degrees Celsius. After cools, the sodium hydroxide is added to adjust the pH, and then the final volume of the emulsion is made up with water for injections. The emulsion form is then homogenized using a high-pressure homogenizer and cooler until droplet size of the emulsion reaches approximately 200 nanometer. This size is necessary to allow the emulsion to be filtered prior to filling process, as larger droplet size might block pores of the filter membrane.
What about the packaging and the closures? How the packaging and closure can be sterilized?
As for the packaging, the container that are commonly used are USP Type 1 borosilicate glass and Type 2 soda lime treated glass. These containers are sterilized through dry heat sterilization as they can withstand high temperature, whereas as for the closure, the rubber closure is sterilized through moist heat sterilizations. This closure must not be permeable to oxygens or become softened by contact with oil phase or emulsions. Finally, the sterile emulsion then is filled into the sterile glass container and seals under nitrogen gas in the filling vessels. Nitrogen gas is commonly used during packaging to maintain the sterility and cleanliness of the product. Not only that, nitrogen can also remove oxygen from the packaging before it is sealed.
Now, let's take a look at the quality control test according to the drug specification.
There are many specifications of the finished products which are needed to comply with the quality control test so that the products are not being rejected and can be released into the market. Firstly, according to United States Pharmacopoeia 32nd edition, identification test is done by ultraviolet absorption or liquid chromatographic system to check for the active pharmaceutical ingredients in the product. For UV absorption, it is to compare the absorbance of test solution and standard solution within spectral range of 200 to 450 nanometer. The requirements are met when the absorption spectra of both solutions show maximum and minimum values at the same wavelength and absorbance ratios are within specified limits. For LC system, it is to compare the retention time and peak responses of the major peak in the chromatogram of the assay preparation and chromatogram of the standard preparation. The LC system should comprise of 275 nanometer detector and a 5 mm x 10 cm column that contains 5 micrometer packing L1 with flow rate is about 2 mm per minute. The requirements are met when the results of both assay preparation and standard preparation are similar to each other. Besides, assay tests also being done by using the LC system. The test use this system specifically, and the results from the peak responses of propofol in assay preparation and standard preparation are calculated to get to know the quantity of API. Thus, the concentration of the API in the injectable emulsion is determined too. Secondly, the bacterial endotoxin test is done to detect or quantify bacterial endotoxins. Gel clot technique is used where specific Lyle reagent is used to be reacted with endotoxins and observed based on the gel form. The gel or clotting will appear if the endotoxin is present. Then, comparisons are made between the test product with USP endotoxin reference standard, and for the product to be accepted, it should contain less than 0.33 USP endotoxin units per milligram of propofol. Next, sterility test is done in aseptic conditions by membrane filtration method and direct inoculation of the culture medium method to check the compliant sterility of the product, and processed in manufacturing emulsion injection by observing the incubated media contain the propofol to be tested. The product is said to meet the requirement of sterility if there is no clear microorganism growth and visually same with the control without product. However, if there is growth in the medium, the experimental conditions for sterility test should be changed, and a validation test is done too to validate the changes made in the sterility test. Apart from that, the pH of the product also being tested. By using pH meter, the pH meter is standardized by buffer used for standardization first before it measures the pH of the solution. For the operation to be accepted, the operational pH have to be almost the same to the theoretical pH of the product, which is between 7 to 8.5. Besides that, the globule size distribution is being checked too, as the emulsion contains dispersed lipid phase in its aqueous phase. By using light scattering method, the mean droplet size of emulsion globules are determined by dynamic light scattering spectroscopy or classical light scattering. For the product to be accepted, the intensity weight mean droplet diameter must be less than 500 nanometer, regardless of the concentration of the dispersed lipid phase in the emulsion injection. In addition, any foreign and particulate matters in the solution are also being measured. The final container of all parenteral preparations are inspected for the presence of observable foreign and particulate matter in their contents with reference to particulates in the visible range of a type that might come out from the manufacturing or filling process by using microscopic particle count test. The test is comprised of suitable binocular microscope, a filter assembly to retain particulate matter, and a membrane filter. The product complies with the test if the average number of particles in the emulsion does not exceed 3000 per container for size that is equal to or greater than 10 micrometer and does not exceed 300 per container for size that is equal to or greater than 25 micrometer. Lastly, the leak tests are done to detect if there is any leakage from the product container or seal. A suitable leak test is done according to different dosage forms. For parenteral dosage forms, various methods are available. One of the tests can be bubble test, where the product container is submerged in a water bath and pressure is applied in the vessel. Then, the leakage can be seen when bubbles or foams are produced when pressure is applied. The product fails the test if there is any leakage is detected. Hence, if the product passed all the tests, then it is ready to go.
Finally, for administration, Propofol Emulsion injection should be administered intravenously. It should be prepared for use just prior to the initiation of each anesthetic procedure. Besides, it is commonly in a ready-to-use formulation and it is used for injection without the need to be diluted. So, let's take a look on how it is administered. The Propofol Emulsion injection are prepared aseptically prior to the administration. Firstly, during administration, this Propofol must be shaped well and it is drawn aseptically into the sterile syringe. When the drug is administered, it will allow the patient to be unconscious before beginning the surgery, and it is also given as continuous infusion to keep the patient unconscious throughout the surgery. There are precautions that must be taken when administered Propofol Emulsion injection. Firstly, Propofol Emulsion injection and any injection equipment like syringe must be prepared for single patient use only. If the Propofol vials and syringe is used on multiple persons, this will increase the bacterial growth as the product may be contaminated. Besides, Propofol Emulsion injection must be administered within 6 hours after the vial was opened. In addition, this drug also must be used within 12 hours after the vials has been opened. Any unused portion of Propofol or solution containing Propofol Emulsion injection need to be discarded at the end of anesthetic procedure or within 12 hours or whichever occurs soonest. Besides, the IV lines containing Propofol Emulsion injection should be flushed every 12 hours or at the end of anesthetic procedure to remove residual Propofol Emulsion injection. So, why 12 hours? This is because this drug can inhibit the rate of growth of microorganisms only within 12 hours. It does not contain any antimicrobial preservative, which cause it capable of supporting the growth of microorganisms after the 12 hours.
In conclusion, Propofol Emulsion injection is used as an anesthetic and sedation drug. This product contains propofol as its active ingredient, as well as other excipients. In addition, it is important to maintain a strict aseptic technique in manufacturing process of this product.
Apart from that, Propofol emulsion injection also needs to undergo several quality tests before it can be released to the market. For the administration step, it is injected intravenously, and each step during the administration must also be prepared aseptically to avoid contamination. Lastly, the main precaution when handling this drug is that it must be prepared for single patient use only and need to be discarded after 12